Well, hi, everybody, and thanks, Mike, for that generous introduction. This is a wonderful time to talk about turning the improbable, a wonderful time to talk about turning the improbable into the possible, as we like to think at Alnylam, and a very opportune moment because we think we're approaching the approval of our first RNAi therapeutic later this year in the shape of patisiran. So let me start with the improbable part of the Alnylam story. 28 years ago, scientists at the University of Arizona were trying to enhance the purple coloration in petunias. And as they tried to do that, the transgene that was overexpressed for the purple color, rather than enhancing the purple color of the petunia, led to absence of color, and they generated white flowers instead of deeply purple flowers. And this particular observation was very perplexing, but through a lot of work, they were able to show that this was, in fact, the first demonstration of RNA interference, and the transgene was leading to degradation of the messenger RNA for the purple color, and hence the loss of coloration and the white flowers. The term RNA interference wasn't coined then. Subsequent work in the 90s in the worm and the flies, and of course the work in the worm by Farr and Mello led to the Nobel Prize for them in physiology or medicine in 2006. But the observation in petunias was reproduced, and the term RNA interference was coined through the observation that the same phenomenon was present in worms and flies. But the first big step towards making medicines out of RNAi came at this institution, which obviously we're all very proud of here locally, Massachusetts Institute of Technology, or MIT, where in Phil Sharp's lab, they were able to show that RNAi, that is the sequence-specific degradation of any messenger RNA in the transcriptome and the ability to silence any gene in the genome, was not only true in worms and flies, but also true in human cells. And once Phil Sharp and his colleagues demonstrated the ability of human cells to execute the RNAi program as part of the endogenous physiology of the cell, it was clear that here was a great opportunity to create an entirely new class of medicines to reduce the expression of pathogenic proteins and tackle a vast array of diseases. And with that, and the backing of local venture capitalists here in Massachusetts, such as Polaris and Atlas, Alnylam was founded in 2002. And our singular mission at Alnylam has been to turn RNAi into medicine. And we want to be leaders in that. But not only that, we wanted to do it in as compressed a time frame as possible. And I say that because if we think about fundamental breakthroughs in biology, and we think back to Kohler and Milstein and the discovery of monoclonal antibodies in the 70s, it took almost 30 years to convert that to the routine production of therapeutic monoclonals. So at Alnylam, not only have we tried to translate RNAi into medicines, but we've tried to compress the time frame we've been doing that in. But as with any new platform, if we look at a fundamentally new technology like this, there are challenges. And there have been no shortage of challenges at Alnylam. The most fundamental challenge for us has been the one shown on this slide here. And that is, if you look at our drug substance, the molecules that we use that execute RNAi, they're termed small interfering RNAs or siRNAs. siRNAs are about 30 times bigger than the average small molecule. So this is not something we could give by mouth for oral absorption. They're also highly unstable and they degrade. And also, they're hydrophilic. They love water. So these are all the kinds of properties that stop our drug substance being a drug-like substance. And the fundamental challenge with these was that we didn't know how to get our siRNAs to a cell type or an organ in the body that mattered. And so we called that the delivery challenge. And the delivery challenge then continued for 10 years from the founding of Alnylam, so from 2002 to 2012. And during that period of time, we had to solve many technical hurdles. And the way we solved them fundamentally involved two things: people and money. Let me go to the money part of the equation with this slide. It's a complicated slide. The jagged lines that you see here represent the Alnylam stock price and the market cap. And you can see over time it fluctuates, although fundamentally over time it eventually reaches a higher steady state. In the middle of this graph, in the period of around 2008 to 2013, which I doubt you can see, there's a very big dip in the stock price. And in those days, no one really believed in RNAi, and we were trading below cash. And what I want to emphasize is that that phase required a tremendous amount of resilience from the company. But as we look at this today, we recognize at Alnylam 3 distinct phases in the life cycle of the company. First was a phase of hope and promise, where we and others were very optimistic we could turn RNAi into medicines quickly. Secondly, there was a phase of fear and despair, and this was the phase when money dried up, there were no believers, we hadn't figured out how to solve delivery, and proofs of concept of RNAi in the clinic were still awaited. And then finally, when clinical data emerged, when in 2012 and 13 we solved the delivery challenge, then you can see the value of the company increases. But more than anything on this graph, the most important thing I want you to look at is in the middle. There's a stepwise line that gradually just goes up throughout the entire time course of the company. And that's the investment in the company. And what we've managed to do, and if there's one big secret on this graph, it's that we've been able to attract investment into the company throughout the life of the company, even during the down days in the middle of fear and despair, such that we could continue to invest in innovation and continue to find solutions to our technical challenges, most notably the delivery challenge. But in 2012 and 13, as we solved the delivery challenge, we started to think about what diseases could we tackle with such a powerful technology whereby we could reduce any pathogenic protein in the body, specifically in the liver. And we turned first to this disease. This disease is termed ATTR amyloidosis. It's an orphan disease caused by a pathogenic protein that's produced in the liver because of a mutation. This misfolded protein then deposits in the nerves and the heart, and patients, unfortunate patients, as you see in this picture, rapidly deteriorate and are dead typically within 5 years of disease onset in middle life. With the realization that we could deliver RNAi therapeutics to the liver, we generated a medicine that we call patisiran today that could go to the liver and silence the pathogenic protein here, the TTR, transthyretin, and hopefully result in clinical benefit. And the very exciting news for us was that in October last year, we were able to release the phase 3 data for the investigation of patisiran in ATTR amyloidosis and showed that we had a profoundly transformative effect on the lives of patients with ATTR amyloidosis in a randomized double-blind placebo-controlled study, the gold standard for investigating a drug. And we were able to show in this study that over half the patients were able to see significant improvements, not just in their neuropathy, but also in their quality of life. And so with that, we filed that drug for approval in the US, Europe, and other territories, and we look to seek approval later in this year. But with that powerful proof of concept in hand now, we've now turned to many other diseases, including other orphan disorders that are life-threatening, like hyperoxaluria, porphyria, and many others. And in the coming years to follow, we hope to realize what we've seen with patisiran and the impact we've had in patients with ATTR amyloidosis in many other diseases. And we think we're well on our way to seeing the dream of seeing 3 approved products by 2020 as a result of this technology, which started with the improbability of an observation in petunias turning into medicines in our company, Alnylam. And as we look at the possibility of making medicines to serve patients, we've also got to manufacture these medicines. And with the help of the state of Massachusetts and the town of Norton, as well as MassBio, we selected a site in Norton, Massachusetts for our manufacturing facility. We're proud to say that we're keeping those jobs local, and scientists from all around the state are coming to help us to manufacture our drugs at this site, which will open in 2019. But earlier on, I said that really the Alnylam story involves science, and that's a fact of nature that we were given the power of RNAi as a natural endogenous mechanism, but we needed a lot of money to get our work done. And then finally, the most important resource of all. And we're very blessed in Massachusetts with all the academic institutions that we have and the many other colleagues in the biotech and pharma system here. And really, what we have done would not have been possible without all my colleagues who I applaud. And so we thank them for helping us turn the improbable into the possible. So thank you for your attention.